Okay, I'll start. Okay, so good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요, and hello all. And welcome and thank you for joining us. The 47th seminar, and I call it on ETH Switzerland Day. As you may know, I had 70 days of seminar trips from May to July, and I had the best weekend in Switzerland with beautiful mountains and lakes. And I would say that nature is sometimes more beautiful than humans, although I met many great people during that 10 weeks. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Professor Mustafa Khammash. He is a professor of control theory and systems biology at ETH Zurich, and I believe I met him for the first time in one of the synthetic biology Golden conferences many years ago. I still remember his great talk regarding integral feedback control circuits for robust gene regulation, and his group experimentally demonstrated his theory beautifully in biological system. In fact, he was a professor in US before moving to Switzerland. So I would say, welcome, back to US, because he's right now in California, Santa Barbara. And Professor Mustafa Khammash, thank you so much for your time and contribution to the community, and the virtual podium is all yours. And thank you.
Thank you very much, Taisuk, and it's a real pleasure for me to be here, to be speaking to you, and also be attending the seminar of Ross Jones. And it's also a good opportunity to be speaking to the young people in the synthetic biology community. But first, let me start by thanking you, Taisuk, for organizing this wonderful global seminar series, which features the work of many young researchers and rising stars. Here we have one such rising star, Ross Jones, who has done really outstanding work on designing genetic circuits for context-independent gene expression. And he will be talking to us also today about stem cell differentiation into T cells. So I'm greatly looking forward to his presentation. So I'm also very delighted to be the 5-minute speaker today. And what I will do with the time is I would tell you a little bit about my own interest in the field, my passion about synthetic biology. And it's really a great time to be working in the field of synthetic biology. In fact, if I was starting all over back in the 90s, I would now know to be working in this exact field. So why do I think synthetic biology is such an exciting field? Well, first of all, synthetic biology is actually a very young field, right? So it's only been a little over 20 years since the first 3 major papers came out in Nature about the feedback circuit, the toggle switch, and the repressolator. And so, 20 years is not really a long time in the life of a field. And so what does that mean to us? Well, it means that there are still many open problems that remain to be solved. And so for a young person entering the field of synthetic biology, there are a lot of interesting problems to choose from. So that's, I think, one reason why I think synthetic biology is really a super exciting field. The other one is that it brings together researchers from very diverse fields and disciplines, from biology to chemistry, physics, engineering, computer science. And this makes it a very highly interdisciplinary field that also has the potential to be intellectually extremely rich field. And I think that's really one of the strengths of this emergent field. And the last reason, or the third reason, is that I think that success in synthetic biology has the potential to have a major impact on society. And so we have the potential, with the right tools, the right approaches, to be able to solve important problems in sustainability, environment, industrial biotechnology, medical therapy. All of these things are within reach from the field of synthetic biology. So because of these three reasons, I think we work in a very, very special field. Now, my own path to synthetic biology was not a direct one. Actually, I started in my research career in the 1990s working on control theory and essentially proving theorems in robust control. And then I got into applications, starting first with power systems. I worked a little bit on power systems. Then I worked on flight control systems with Rockwell Collins. And then by the late 90s, early 2000s, I, by chance, happened on a problem in biology that's related to how our bodies regulate calcium, so calcium homeostasis. And then I was hooked. So gradually, my research switched from electrical engineering and control theory to looking more and more about biological problems. In particular, we were looking at stochastic systems and stochastic dynamics in biology, developing tools and methods that allow us to characterize and analyze systems noise in biology. Now, at that time, it was purely computational work, and it was mostly systems biology. It was kind of reverse engineering biological complexity. But as an engineer, I was itching to work on design. So control engineers want to design things, and so we would like to look at forward engineering. And the opportunity to do that came in 2011 when I moved with my group to ETH Zurich, and that's when I started my wet lab. And this was the opportunity for me to actually do forward design in biology, and that's basically synthetic biology. So that's when we got started, and I think it's been an amazing ride since. I enjoy every day I go to work. I think it's a lot of fun to go to the lab and just try to marry theory and practice together in order to solve important problems in synthetic biology. So I thought maybe in the last couple of 3 minutes, what I will do here is I will looking back at how my career started and how I switched different fields. Maybe I can give a couple of pieces of advice to young PhD students, but also young researchers starting in the field, at least from my own perspective. The first one has to do with the importance of the problem selection. When you select your PhD problem, or you select what problem you would like to be working on as an assistant professor, and you'll be spending a few years working on this problem, it's really underappreciated how important the problem selection is. And here I would like to quote from a scientist from Bell Labs, a famous scientist by the name of Richard Hamming, who had designed the Hank filter and has many other contributions in communications and signal processing. So Richard Hamming wrote really a marvelous article on doing research. And let me just quote one of the things in this article. He says, most scientists spend almost all of their time working on problems that even they admit are neither great nor are likely to lead to great work. And then he says that if you don't work on important problems, you are not going to do important things, except by the dumbest of dumb luck. So I guess the point of this is that one should spend enough time to find those important problems and work on them. And our field has many of these. One good example is the problem that Ross is going to talk about today, which is stem cell differentiation, among many others, of course. The other has to do with actually interdisciplinarity. So as you know, much of the exciting work, much of the exciting research, not just in our field, but also in other fields, happens at the boundary of disciplines. And so while one is most comfortable working in one's own discipline, I think it's important to stretch into other neighboring fields and to have the courage to kind of unshackle yourself from your own disciplines and try to learn neighboring fields and try to make connections with people in those neighboring fields. Because after all, the solutions to the big problems are not likely to come from a single field and will likely involve methods and tools from several fields. And so it's important to be prepared. Of course, you have to be an expert in your own field, but you should also have knowledge in neighboring fields. So if you're a theoretician or a modeler, you should learn about the experiments and how experiments are done. And if you're an experimentalist, you should take the time to learn about the developments in modeling and theory, machine learning, and so on. And even though it may seem irrelevant at the time, I think you will find that these things that you have learned will come back and help you one day. The third thing I think has to do with my own experience with the role of theory in synthetic biology. You know, I think that in my experience, every field needs a good theory. It needs a good foundational theory. And synthetic biology is not an exception. And so why would we bother with theory? Well, I think theory allows you to kind of abstract the main concepts and then study them in isolation from the specific instances and be able to make more progress at this abstract level, and then eventually come back and apply these abstractions to the specific instances. In our work, it has been extremely powerful, extremely useful. And in fact, without theoretical considerations, we would not have been able to come up with some of the designs that we came up with. And here I would like to quote somebody else, Kurt Lewin, who basically says, there's nothing more practical than a good theory. And so don't do theory for theory's sake, but I think a good theory will ultimately be very practical. So if you are a theoretician, I urge you to embrace theory and be ready to forge new tools, not to just use the tools that you already have to fit the problems in synthetic biology, because synthetic biology brings in its own problems and therefore requires the development of novel theories. So one should be willing to do that. But I think the reward is quite substantial. And finally, I think it's very important to have fun while you're doing your work and to enjoy the ride. And so with that, I'd like to leave the podium to Ross. Ross, I'm really looking forward to your presentation today and very excited to learn what you've been up to since leaving MIT.
Wonderful, wonderful. You know what? I agree every single point you mentioned. Also, you summarized very well about synthetic biology. As we all know, synthetic biology a little bit unique because synthetic biology at the beginning combined mathematics theory with the experiment, and that two, whatever three paper you mentioned, basically start the field. And then the keyword you mentioned is interdisciplinary and collaboration that make the big impact to the society. That's why I'm super excited with this field, because scientists should give something back to the community and society. And then I believe engineering concept of this field basically allow us to do that. So I'm really thankful you mentioned everything beautifully within a very short time, and thank you so much for your advice. That's invaluable advice. Thank you.
Yeah, thanks.
Okay.
Thanks, Mustafa.
All right, so now the main speaker of today with a longer introduction. And Dr. Ross Jones is a postdoctoral fellow at the University of British Columbia, working in stem cell bioengineering lab. And I love, UBC, beautiful Vancouver and beautiful ocean, mountain, everything they have, as well as science and engineering over there. His research focused on applying synthetic biology principles to program development of stem cells into immune cells in vitro. He earned his PhD at MIT, working under both Professor Ron Weiss and Domitilla Del Vecchio. I mean, both of them I love. And there he studied resource competition among heterologous genes in mammalian cells and applied control theory principles to improve the performance of genetic circuit in varying intracellular context. And that's not easy kind of topic, very challenging one, but he successfully demonstrated beautifully. His PhD work, I believe, is aligned well with Mustafa's work, and Ross is a true rising star with both mathematical and experimental skill set in even 2 countries. I'm talking about Canada and US. I may need to say that today is a global exchange day because both speakers today are brave enough to move to another country and did their important work in 2 countries. Of course, the impact is global. So Ross, take it away, and thanks so much for your time and contribution to the community. And I believe if you are an employer potentially looking for faculty position, filling, I believe Ross will be one of the person you need to, chasing. Otherwise, Ross will be in another your competitors' university. So thank you, and it's all yours.
Thanks, Taeseok. That's really nice. All right, let me share my screen. Can you confirm that you can see it?
Yeah, so wonderful.
Okay, awesome. So also thanks, Taeseok, for talking of Vancouver. Here's a nice picture of it to encourage people to come visit, come check it out. This here is UBC, which is on a peninsula. It's just a beautiful place, mountains right nearby. It's a great place to work on science, and I'm thankful to my current lab for taking me in and teaching me a lot and allowing me to bring in my synthetic biology to the lab and help develop that within the context of trying to program T-cell development. So to get started here, let's talk about T-cells for the people that watching this that might not know too much about them. So there are 2 main types of conventional T-cells in the body: cytotoxic T-cells and helper T-cells. The cytotoxic T-cells are what hunt down infected cells and cancerous cells and other cells that have problems. And helper T-cells help to regulate the immune system, especially when it comes to B cells, which produce antibodies, such as the antibodies that we need to fight COVID. The way that T-cells work is that they express this large complex called the T-cell receptor, which includes other key proteins called CD3 in the complex. The T-cell receptor recognizes peptides presented on other cells. And when the T-cell receptor, which for each T-cell is specific to one peptide, recognizes that peptide on another cell, it induces the cell to proliferate, release cytokines, and in the case of cytotoxic T-cells, kill the target cell that was presenting that peptide once the cell is in an active state, or once the T-cell is in an active state. So we can reprogram T-cells to recognize any antigen that's presented on the surface that we want, an antigen being some sort of protein that we want to recognize by putting in what's called a chimeric antigen receptor. So chimeric antigen receptors are basically pieces of antibodies stuck together with something that looks like a T-cell receptor on the inside. That also includes a co-stimulatory domain that kind of packs all in one the punch needed to activate a T-cell once it recognizes some protein on the surface of another cell. And it doesn't have to be a protein fragment like the T cell receptor, but it can be any full-length protein of interest. The kind of first and most widely recognized example of this is targeting CD19 on B cell lymphomas. CD19 is a B cell marker. And so you can basically use CAR T cells to destroy all of the B cells and thus cure the B cell lymphoma, which is a type of cancer. The way that the therapy version of this works is that we extract T cells from a patient. We would engineer them in the lab with either a viral vector or some other means to express the CAR. We would then expand those cells and administer them back into the patient. But of course, this is a problem because this is extremely expensive and also it takes a lot of time to do outside of the patient. Some of these therapies can run upwards of $1 million. So how can we reduce the cost? Well, one way that you could feasibly get around it is to produce the T cells totally in vitro without the use of a patient. So for example, if you had human pluripotent stem cells, which are a type of cell that can be differentiated into any other kind of cell, and you engineer those with, let's say, a molecule or the capability to express a CAR, you could clonally isolate a line, which is very nice from a regulatory and manufacturing perspective, expand those cells, and then differentiate them into T cells. But of course, the latter is easier said than done. In this way, we get around the need to get cells from donors or from patients themselves, cutting out a lot of the cost and time in the process. So our lab has been doing just this over the last several years, and there's been some really great work led by people shown up in the right corner in our lab, headed up by Professor Peter Zandstra. So we've set up a process where we can take pluripotent stem cells, differentiate them towards what are cells that are similar to blood stem cells and are capable of differentiating towards T cells. On this path, they first differentiate towards a cell type called pro-T cells, which are an early committed form of T cell progenitors that express these two key markers, CD7 and CD5. Those then further differentiate into what are called double positive T cells that express both CD4 and CD8 instead of just one or the other, as in the case of mature cells. Here's an example of some of the cells from our differentiations expressing both of these markers and thus are at this stage. And then finally, we can differentiate the cells further all the way towards cells that have the TCR and the CD3 complex and express either CD4 or CD8. And so they've matured fully into T cells. Most of the cells that we make, as with most other labs trying to make T cells in the lab, are CD8 single positive, which means that they're the cytotoxic form of T cells. It's been a general problem to make the helper T cells or the CD4 positive lineage, but that's a topic for another time. So the early parts of this differentiation are critically regulated by signaling through the Notch receptor, which is why back in 2017 or so, Peter and Shreya, who was initially working on producing pro-T cells from blood stem cells, founded Notch Therapeutics. If you're interested in all of this, they're hiring, so check it out. The later stages of the T cell development still involve Notch to some degree, but there's a lot of signaling that goes through the T cell receptor as that receptor gets produced and put onto the surface of the cell progressively in a complicated stepwise fashion that I won't get into. Okay, so the confluence of Notch and TCR signaling is very important for this process. And it's actually a little bit more challenging to make a CAR T cell than you might assume. So for example, there is some very recent work that published a iPSC to T cell protocol that was looking just at this. What would happen if they put a CAR into the cells and then differentiated them? So for their unedited cells, they can differentiate them to the CD4, CD8 double positive state, the sort of intermediate maturing form of developing T cells, but not fully matured. When they put in a CD19 targeting CAR, which is again the most widely and commonly studied form of it, it basically prevents them from going to this double positive state and sends them towards alternate non-canonical like fates. It's still capable of some functions of T cells, but not all of them. But when they delay the expression of the CAR or mutate it so that it's less strong, then they're able to recover some of the normal T cell developmental progress. They think that based on their measurements of the several genes in the cells at different stages of differentiation, that the expression of the CAR is basically suppressing signaling through the Notch receptor, which, as I mentioned, is very important for pushing cells through the early stages of T cell development. And so there's sort of antagonism going on here between trying to put in this therapeutic modality and then trying to differentiate the cells to T cells, which is frustrating. But just to throw another wrench in this, we've been also working on producing CAR T-like cells from our system. And to show you an example of a different CAR that we were looking at that recognizes a different antigen, when we put this into the cells and differentiate them, it doesn't really affect their development towards the protein lineage very much. We think that the differences between these probably has to do with different basically potentials for basal, or what is referred to in the CAR T world as tonic signaling, which is just the signaling that is coming from this and activating the cell, even when there is no actual target that is being recognized. And the CD19 CAR is known to be quite high in tonic signaling. Further, we think that this tonic signaling is then affecting the differentiation outcomes because stronger Notch or TCR signals are more strongly affecting the expression of Notch ligands. So this is something that we'll continue to study and investigate in the future, but it helps to set us up with a sort of mental image where we have Notch signals, we have TCR signals, we also have all of these factors that we've defined in the media, because we have a fully defined system. We know everything that's in the media, at least when we put the cells into the media. And but the problem is that all of these inputs that we provide to the cells are going into what are actually in human cells pretty poorly understood gene regulatory networks that drive the differentiation of the cells. And as demonstrated with this CAR introduction, these GRNs are feeding back onto these signaling pathways that we're trying to regulate with the culture system that we set up. So when we introduce a CAR, it's perturbing this system, and the effects of that can be unpredictable. This is further complicated by the fact that at any given time in the differentiation, there can be various cells at various stages of differentiation, all secreting things that might perturb every other cell. And so we have to be very mindful of what's going on in the culture as an entire system. It's not just individual cells all at the same state, all reacting the same exact way. So this sort of way of thinking about the signaling inputs and the state of the cell can be helpful to pitch this into the frame of reference of context dependence. So when we're introducing something like a CAR or an engineered TCR, which is another type of therapeutic molecule we can make, or other factors that we can put into the cell, either for therapeutic purposes or to help drive the differentiation, these are going into this environment where there's a lot of feedback effects and we don't know everything that's going on. Further, for the things that we put into the cell, we have to consider the genetic context of where they're going into the genome, how they're composed themselves. We have to consider how the cell state is changing over time and how all of the confluence of transcription factors and other factors that are involved in gene regulation are going to affect the production of these genes and their ability to sustain expression over time. And finally, as I talked about before, the extracellular context, all of the signal inputs that we provide, and all of the inputs coming from other cells become important for affecting the whole cell and the genes and such that we put into it. But our genes that we put into the cells can further manipulate this all of these contexts. One way to simplify this a bit is to say, think of context dependence as a disturbance input. So let's take a cell here that we've engineered to do something. It's taking some inputs from the environment and then producing some outputs in response. Let's say we've introduced some sort of gene, which we can call a genetic device, that itself is taking inputs and outputs from within the cell. This gene is comprised of a DNA sequence that's producing some mRNA and protein. These mRNA and protein species we can consider to be the outputs of the device. And those outputs are what are affecting the cell state if this is a device that we've programmed to, let's say, drive differentiation. So the inputs that we are providing to this device, or that we want it to respond to in the cell, we can refer to as the regulatory inputs. And then all of the context-dependent effects that are going to affect the production of this gene and its function in the cell, we can consider as the disturbance inputs to the system. And so this allows us to kind of just simplify how we frame and think about the context-dependent effects, especially when it comes to modeling, which has been really useful for us, and I think for Mustafa's group as well. So all of these inputs affect the different production and decay parameters in the system. And so by accounting for them, we can be a little bit more aware of what's going on and try to design systems that are aware of these disturbance inputs and can operate even if they exist or might be changing in ways that we don't know. So one way to design systems that are robust to these types of disturbance inputs is with feedback control. And definitely check out Mustafa's work if you're interested in this. But I'll talk a little bit about what we've done on this front. So a feedback controller is a device that detects the output of some device that we've made of interest, compares that output to what it wants the output to be based on the level of input provided to the controller, and then actuates a response so that the device output will match what the controller has specified it should be in response to the input. So when there are disturbances coming from cell context or other factors like stochasticity and gene expression that are plugged into this device, then the controller can detect changes in the output and respond. So what we'd expect to see is that, let's say, when there's a disturbance that comes in, the controller actuation will pick up, and this will return the device output to some predefined level that was specified by the input. So if the system is robust, then basically it has the ability to reject disturbances. So we like to be able to design very robust systems that don't depend too much on their context. So we think that feedback systems could be particularly useful for programming development because we could use them to detect the state of the cell and then provide inputs back into the cell state to sort of adjust the differentiation progress. In this way, we don't have to necessarily try to optimize every signal that we're putting in perfectly. We just have to look at what is the output of those systems with respect to things that we can measure in the cell, and then we could feed that back in to the controller to update the state of the cell based on what we're seeing. And so this is still sort of a very much work in progress vision, but this is what I'd like to be able to get to over the next few years. So for today, what I'm going to talk about is an example of a feedback controller that I worked on in my grad school work under Ron Weiss and Damatilla, with great input from Yili Kian, who's a grad student at Damatilla's lab now at University of Wisconsin. And I'll also talk a little bit about, once we have control systems that could feasibly control gene expression and help to control differentiation, how we think about putting those into the cells and composing them in ways where they'll be where the individual genetic components will be stably expressed and expressed strongly and sufficiently to be able to drive the types of programs that we need. And a lot of back and forth on this has been going between me and Katie Galloway and members of her lab who have been really interested in studying this context-dependent effects at the genetic level as well. Okay. So way back early on in my PhD, we were thinking about ways to deal with resource competition. And actually the first way we tried was before we got into some feed-forward control work that ended up getting published first, was this design that Yili and Domitilla worked up, where you could have a covalent modification cycle where a kinase and a phosphatase would reversibly phosphorylate a gene regulator. And that gene regulator in its phosphorylated form would regulate the production of the phosphatase, probably through activation. If you had the regulator also produce some output protein, then the kinase would serve as the input to a system driving the production of the output. If you have a large amount or concentration of this gene regulator, both in the phosphorylated and unphosphorylated form, you can saturate both of these enzymes, which mathematically leads to a result where you can get perfect control of the level of output based on the amount of the kinase input through integral-like control, which I won't go into detail here, but I'll provide a reference to Yili's paper if you'd like to learn more. So if this works well, then it should be robust to any perturbation that comes within the loop between the kinase and the output. And the nice thing about phosphorylation reactions is that they're very fast, which means that we can have a strong feedback gain even if we don't have that much of the phosphatase protein being produced. And Yili showed that this strong phosphatase activity is necessary in order for the system to act robustly and to be able to protect the output from perturbations that would be input to the system in a way that is comparable to some idealized version of this that isn't operating in a cell that has a lot more problems like cell division to deal with. So the way that we implemented it was by turning to two-component signaling. And this is a bacterial form of signaling that's not really present in animals, which is really useful for us because there are so many signaling pathways in human cells that to imagine trying to engineer kinases and phosphatases in this space would be fraught with orthogonality issues where we might mess up the existing signaling pathways in the cell. And so we thought, let's take a signaling pathway from organisms that will be completely orthogonal by default to those in the cell because this type of signaling doesn't even exist in animal cells. So the way two-component signaling in bacteria works is that we have histidine kinases that sit in the membrane and detect signals either in the periplasm of the bacterial if they have two membranes or in the extracellular space. When the signal is seen, then the histidine kinase will autophosphorylate on its histidine residue, and that phosphohistidine will then transfer its phosphate group down to an aspartate residue on the cognate response regulator protein for the histidine kinase. And most of these response regulators are transcription factors, which is really useful for us because then we can use those to drive gene expression in cells. Many of the histidine kinases are also bifunctional. So in the absence of signal, they act like a phosphatase and strip the phosphate group from the response regulator. And when they receive input, then they get switched to a kinase-like state where they're then actively transferring the phosphate group onto their response regulator. And so this means that there's a potential way for us to extract individual kinase and phosphatase proteins from a single histidine kinase. There are dozens of orthogonal pairs of histidine kinases and response regulators in E. coli alone. So there's a huge set of potential parts that we could use to implement these types of feedback control systems and other artificial signaling systems in human cells, which I'll also point people to Kobe Benenson's lab and others that have been studying these types of systems and their ability to transmit signals in human cells. Okay, so the ones that we chose to study is a pair of proteins called ONSI and ONBAR, which are the most well-studied two-component signaling proteins in E. coli. And it's very helpful that they're well-studied because we can use a lot of the information that's known about them to engineer them. And I'll skip a lot of the details behind how we engineered them for the sake of time. But at the end of the day, we came out with a protein that had the phosphatase activity mutated to remove it. So it kind of acts just like a kinase, and another protein where after a lot of effort to make a good phosphatase, we finally found an architecture that worked, where we truncate the ONZ protein and then affix these leucine zippers from the GCN4 protein from yeast, and got it into a conformation that finally, after really a long time, gave us really strong phosphatase activity. And also note that this includes a mutation to knock out kinase activity. All right, so we use these proteins moving forward. Check out the paper if you want to learn more about how they're derived and what they look like. So, all right, so we plug them into our feedback controller, which at a box level looks like this. We have a kinase feeding into the phosphorylation cycle, which we consider technically to be the controller element here, which drives transcription of the output protein and also a phosphatase. The phosphatase stability is a parameter that we can tune through a small molecule input to the system, which allows us to tune the feedback gain of the circuit, which is very useful for looking at certain properties. So what it looks like when implemented is this. We have the protein en bar, the response regulator here fused to VP64, which is an activation domain that allows this to work as an activating protein in human cells. When we add the kinase variant of ONZ, then it phosphorylates this protein, and that activates production of both the output and the phosphatase. The phosphatase protein is fused to these degradation domains called DDD, so that the phosphatase, when it reverses this phosphorylation reaction, can be degraded with the small molecule trimethoprim to tune that dephosphorylation rate. An open loop version of the circuit that we tested in parallel replaces the phosphatase with a luciferase, which is inert with respect to the phosphorylation of ONPAR, and thus serves as sort of just like a nothing protein. It breaks the feedback loop. Okay, so we took this system and we tested its ability to respond to perturbations. And what we predict, just in general, because it has a negative feedback loop, but specifically based on the model of the system that Yili composed, is that as we perturb the production of the output in the phosphatase, the closed loop system that with the feedback control compared to the open loop system should be able to suppress any effect of the perturbation and keep the output level closer to what it should be as specified by the amount of kinase. So we tested out two perturbations to see how this worked. One is a microRNA where we put a target site for it in the 3' ETR of the gene. MicroRNAs, there's hundreds of them in the cell. They're small RNAs that target and regulate gene expression. And they're a good model for the changing cell state during something like differentiation, actually, because there's all of these microRNAs and their expression changes over time to regulate genes during development. And it's certainly possible that any gene that we make will have cryptic sites for these microRNAs that can affect their production and expression. The other perturbation that we tested was the expression of a protein called Galford Viper. In a previous paper, I showed that this is an extremely potent transcription factor that is actually so strong that it suppresses transcription of other genes. And so it's a good model for the changing suite of transcription factors in the cell in general, but also the interrelated effects of different genes that we express on each other when we put them together into a circuit. But together, these perturbations give us both a pre and post-transcriptional perturbation. And so we can confirm that the circuit works for perturbations both at both of those levels, which it should be able to handle if it's working. So first we'll look at the response between the kinase going in and the output on the other side. Shown here are a couple different open loop configurations in red. And two different closed loop configurations in blue and green. The blue one doesn't have the destabilizing domain onto the phosphatase. It's just the wild type, or not the wild type, but it's just the phosphatase by itself. And what you can see is that both of the closed loop versions of this really strongly suppress the ability of the kinase to turn the system on. And that's probably because the phosphatase is actually maybe too strong, but that's all right, because the destabilization domain and using it in a regime where it's destabilizing the protein quite a lot allows us to recover a lot of the dynamic range that we would expect to see from the open loop configuration back to a more reasonable level. So we use these two different open loop variants because they have, or for the sake of simplified comparisons here, because they have similar initial levels of output when there's no kinase. But in the paper, we compared systematically a lot of different variants of both the open and closed loop systems with different starting and final levels so that we could have a systematic comparison of different output levels at different kinase levels. So in the absence of kinase, when we add these two different perturbations to increasing levels of perturbation shown on the x-axis and measure the fold change in the output here on the y-axis, we can see that when there's no kinase added into the system, the response of these to their sort of paired control is very similar. But when the kinase is at the top of its range from this experiment, then we have the ability to sort of recover from the perturbations, especially in certain operating regimes. It turns out that the Galford Viper perturbation was so strong that we ended up basically returning these systems back to their baseline when it was at sufficiently high levels. But when it was at lower levels, we had a much better ability to sort of control the response and keep the output closer to where its baseline was. So if we look at the kinase input versus the fold change in the output for just one level of perturbation of the microRNA in Galford, you can see that the feedback controller performance improves, especially relative to the open loop system as we add more of the kinase in. And this makes sense if you think about it, because when there's no kinase in the system, then there's no phosphorylated transcription factor to dephosphorylate. And as we add more and more kinase, then we'll generate more and more phospho on bar, which then will help to push the phosphatase towards the saturation regime where it works more effectively in the feedback control based on the mathematics of this. And also note that we looked at many different open loop variants here, and just to confirm that it wasn't just the fact that the expression level of these genes is lower in the high kinase state than it is in the open loop systems that's causing them to have less fold change. It is like a real effect in this sense. Okay, so another benefit of this that we found is that when we induce the gene with the kinase, so now not really looking at the perturbations anymore, there's a very broad and kind of noisy transition in the distribution of output from off to on for the open loop systems, here showing the same two that we were looking at before. But with the feedback control system, we can tighten up those distributions and give a much more uniform unimodal shift from off to on as we induce the output with the kinase, which is what we'd expect if the feedback control system is working, but it is a nice confirmation again that the feedback control can basically actuate a more robust input-output response than the open loop system. Okay, so kind of with this in mind, we can sort of imagine that with feedback control systems, we could use them to take all of these different inputs that we're providing to the cells during something like differentiation, for example, stimulation through the Notch and TCR receptors. And we can take all of the synthetic genes that we're putting in that are supposed to be helping to drive differentiation, or that we just want to be there for therapeutic functions once the cells finish differentiation. And with the controller, sort of sense the outputs that are coming from each of these genes, detect what they're doing, detect the state of the cell, and then use that to regulate the cell fate and hopefully drive it towards the T cell fate or whatever we're trying to get the cells to differentiate into more robustly. And in this way, we're not just saying that we want to, let's say, control the expression level of a CAR or something that we're putting into the cell, but we want to detect the effects of these genes that we're putting in and the signals that we're providing to the cells on the state of the cell, and then use that to update those inputs to optimally drive the differentiation. And so this becomes more of like a, instead of controlling one gene, moving towards a system where we would be trying to control the phenotype of the cell more broadly. But again, this is work in progress. So stay update, or if you're interested, keep aware. Hopefully, we'll be coming out with things related to this over the coming years as I continue on in my postdoc. But something that I can talk about right now is something related to the genetic context of things that we're putting into the cell. So if we put a controller into the cell, if we're putting in these synthetic genes like CARs, these are going into a cell that's differentiating, and the context of how we introduce them into the genome itself can be very important for getting them to function properly. We don't want to put a controller in and have it get silenced after 10 days of differentiation, and then it becomes completely useless. So for that, I'll talk about some things that we've been noticing lately as we've been going through and engineering stem cells and differentiating them. So one thing that has really been kind of almost shockingly strong of an effect that we noticed is that depending on the orientation of two genes that you put together into a circuit, so these are integrated into stem cell or pluripotent stem cells in a piggyback vector. If you have, let's say, a PGK-driven reporter in the first position and a HEF1A-driven reporter in the second position, or swapped, it dramatically affects the relative expression of the two markers in a way that was far beyond anything that I had sort of expected to see. And yeah, it surprisingly to also me, somebody who thought that I knew something about gene insulation and how promoters and enhancers might interfere with each other through my study of resource competition and the resources that promoters use, putting in insulated region or insulators to sort of putatively block these promoters from interfering with each other didn't really have much of an effect on the relative expression of these two markers. And so there's something else going on most likely. And I thought that I would reach out to Katie Galloway's lab because I knew that she was studying some of these context dependent effects as well. And she had been looking at something called DNA supercoiling. So the effects that we see are consistent with a DNA supercoiling model. And to give you an example of some data that Katie sent me and has presented about recently, we can take a look at two gene configurations where you have an inducible gene either in a divergent orientation with a downstream gene or in a tandem orientation so that they're both aimed the same direction. And what Katie's seen and what other groups that have studied DNA supercoiling have seen when you have these integrated onto a linear piece of DNA is that in the divergent orientation, when you induce the backwards gene, you get sort of an increase in the expression of the downstream gene. And when they're in a tandem configuration, the induction of the upstream gene suppresses the expression of the downstream gene, so that you can end up with actually quite a large difference in what the initial to induced state of the second gene is, even if you're not trying to do anything to it specifically. And so this is referred to as upstream dominance, where when you have two genes next to each other, the first gene in tandem, the first gene's expression will dominate that of the second gene, and is basically exactly what we were seeing in our experiments, and the reason why I reached out to Katie. The results that they see in this experiment is also really consistent with some modeling work that Christopher here did. I definitely encourage you to check out his paper on bioRxiv, where they went into great detail on studying not just the kind of average expression changes in response to different orientations of the genes, but the stochastic and sort of transcriptional burst kinetics that would be expected for genes in different orientations and on different things like a plasmid versus integrated into the genome. So that helps us to explain a little bit about what's going on, but we're still seeing things that we don't quite understand. So for example, if we're looking at a HEF1A driven tag BFP reporter and looking at what happens to it when we add different downstream genes with different promoters, you basically see that the expression level of the BFP reporter is not that much affected by what's downstream of it. maybe a little bit higher when CAG is there, possibly, but through sort of activation, enhanced activation of the HEF1A promoter by the extremely strong CAG enhancer. But when we put different promoters upstream of the HEF1A gene, this is the circuit that I showed before, where we had PGK, or very similar to the one I showed before, but not exactly the same, where we have PGK promoter driving the upstream gene. This strongly suppresses production of BFP from HEF1A. When we have the CAG promoter in the upstream position, then the BFP reporter becomes kind of intermediately high. And when we have HEF1A upstream of HEF1A, then it's robustly expressed, basically the same as it was when it was the upstream gene. So the upstream dominance phenomenon that we've observed so far that is consistent with DNA supercoiling also has something else going on that we don't quite understand that depends on the promoter itself. And so we're hoping to learn more about this basically soon, and get a better idea basically of how and why different promoters would interfere with each other's ability to transcribe, and hopefully update the model that Katie has been developing in a way that helps to account for these different behaviors. So yeah, a lot more to learn, but we can already see how some of this translates into differentiations. So the original impetus for looking at the gene expression differences is because the first one that I had made was one that had PGK and HEF1A together. And in PSCs, I saw this big drop off in expression of HEF1A because it was the second gene in this sort of cassette, whereas the PGK was stable. When I differentiated these cells towards pro-T cells, the neon green signals stayed not super high, but there was a reasonable number of cells left over that were still positive for BFP, but there's basically nothing after this length of a time differentiating that still had the BFP reporter. And so I thought, oh no, like the HEF1A promoter is not stable through this differentiation. But after testing all of those different configurations and realizing that there was this upstream dominance phenomenon going on, I later designed another circuit where, well, this was a control for something else, but basically it had a neon green reporter in the upstream position driven by HEF1A, and also a selection marker downstream of that. After selecting the cells with this integrated via piggyback, then we get nearly all of the iPSCs expressing the GFP reporter. After then differentiating them for the same amount of time as here, but without selection, we can see that a good number of the cells are still expressing this gene. So it's not that HEF1A was unstable in this first system, it's just that the orientation of the gene was suppressing the ability for it to be expressed. So now we can sort of imagine how being aware of this context helps us to design systems that will be able to be expressed reasonably reliably during the differentiation, and to then be able to accurately control the outputs of the signals in the cells so that we can regulate their development. So yeah, it's been fun to kind of look at these different effects with Katie, and we're hoping to learn more about it so that we can reliably program development. So in summary, I've talked about how PSC-derived CAR T cells could be a great alternate to the current way of doing the CAR T therapy. So we don't have to get cells from patients, but they're challenging to make. Context dependence affects all of these aspects of programming cell development, and we have to be aware of it in order to more robustly design systems that will interface with the cell. And control systems can be a really nice way of taking these context-dependent effects and trying to minimize their effects on gene expression generally, but development specifically. So with that, I'll say thanks to everyone that I've worked with across multiple labs over the years, both at MIT and at UBC. It's been a lot of fun. Thanks to my undergrads and all the people in the Zanstra lab that have helped me out in getting started on my postdoc and in sort of contributing to everything that I've done, including preparing for this talk. And the last thing I'll say, for all you grad students and postdocs out there that are watching this, as a membership outreach committee co-chair on the Student and Postdoc Association of EBRC, it is my duty to try to get you to sign up for EBRC's Student and Postdoc Association. We've got a lot of great programs for you. workshops, networking events, internship programs where you can get connected with academic or with industry members that you might be interested in working with in the future. We've got mentorship program that you can get connected with great mentors in the field, and outreach programs where we can try to reach out to college and high school students to evangelize the field of synthetic biology and engineering biology more generally. So use this code to go find the application form. It's short. If you're doing anything related to synthetic biology and you're a student or postdoc, grad student or postdoc, then we will get you on board. All right, so that's it for me. I'll turn it over to questions.
Yes, join EBRC SPA today. Don't delay. I mean, today, right now. Okay, wonderful talk. I love it. I have actually many questions, but I wanted to ask audience and Mustafa, if you are still there, you could ask challenging question or comment.
Should I stop the screen share and go
it's
back
up to you. It's up to you.
Okay. I'll keep it here for a minute in case you guys want me to bounce back to anything.
Sure. Mustafa, if you don't have, I will start with one person raise your hand. I will, or you can start, but I have Teresa. I will allow her to talk. Hey, Teresa.
Hi, Ross. Hello.
Hey, Teresa. Good to see you again.
Good to see you. Awesome talk. I was wondering, and maybe I missed this to some extent, but so this effect that is not fully explained by supercoiling, but is like specific to specific promoters and like the relative to promoters, is it possible that it's just about promoter strength, or is it definitely like something even more spooky than that?
Yeah, well, the weird thing about it is that based on the expectation basically from supercoiling is that stronger transcription upstream will suppress transcription downstream. And I didn't talk about the detail of this, but for the people that might be listening to this that don't know much about supercoiling, so when transcription is coming upstream here, it'll induce supercoiling in the DNA that should prevent RNA polymerase and other transcription factors from binding to this promoter effectively and transcribing it, which, yeah, when we have this case here is kind of the opposite. So HEF1A is a stronger promoter than PGK in PSCs. And so we're seeing kind of the opposite effect than what we would expect. But nevertheless, it could still be a strength or like at least a frequency-based thing. So one thing that we've been thinking about is, does this work well because both of these promoters are operating at the same frequency? Or is it, you know, some specific effect where PGK and HEF1A don't play nicely? There's certainly like more to learn about how insulators might help here. I've looked a little bit at that as I showed, but you know, that it doesn't mean that we've fully stopped any sort of 3D interactions between these genes that might be sort of antagonistic from happening. So it could be something just related to that. It could be the relative strengths not being matched. That's not ideal. But we have seen that when we put the same promoter in tandem, not just HEF1A, but CMV-based promoters and other ones, that they operate basically fine.
Wonderful. So before I ask Mustafa asking or comment, Teresa is one of the early speaker in this series, and she got a job, you know, faculty job, and congrats, and you know, welcome again to the. So
thank you.
And then another thing I want to say to Ross, I mean, you did fantastic job. You know, your talk is very robust because there are so many noise. I mean, background noise. You know, fire, whatever.
Oh,
you know.
I know. Sorry, I'm in downtown. I probably
downtown. I know. I'm in Vancouver. I know very well. So, but you did a great job. So, Mustafa, you have any challenging question or comment?
Yes, I not necessarily challenging for Ross, but just curious. Very nice talk, Ross. Really beautiful presentation and nice work. So I just want to ask about the phosphorylation circuit that you showed earlier.
Mm-hmm.
So if I remember correctly, in principle, this circuit should be functioning or should be capable of functioning as an integrator and giving robust perfect adaptation. But usually the circuits that are supposed to function as integrators in practice, they face several challenges that make them less ideal as integrators. And in this case, I think this is one of those scenarios.
Yeah,
so
exactly.
What, in your view, is the biggest challenge that keeps you away from being in the regime where perfect integration is actually taking place?
Yeah, thanks for asking, because not something that I had too much time to go into. I'll bring the circuit back just so we can take a look at it. The key property, right, is that we need to saturate both of the enzymes. And mathematically, this makes it so that the change, or basically the reaction rates, become proportional to the enzymes and independent of the amount of the substrate that's there. So the zero
you need
order
zero order reaction. Yeah.
Right. So we need zero order reactions. Well, I forget off the top of my head if we need it for the kinase. I think we need it for the kinase for it to be linear between kinase and output, but we need it for sure for the phosphatase, because we need that feedback to be linear. So we think that what happens here is actually that even if we were to get into that regime, the problem is that the AMPAR protein, in order to get that high to saturate the phosphatase when it's in its phosphorylated form, it would also saturate the promoter. And so we're kind of either saturating them at a similar time, or we're saturating the promoter first, which is a problem. So because then your phosphatase level can't even change in response to the output there. So there's this sort of key two parameters that we realized probably too late to fix this before I graduated. The KD of binding between the phosphorylated AMPAR and the output promoter, and the Km of this enzyme coming here. And you want the Km to be as small as possible relative to the KD, so that you can saturate that first before you saturate the promoter. And we think that they're similar, or maybe even the KD is smaller, because a lot of these proteins have pretty small KDs for binding the target promoters. And actually a lot of enzymes have high Km so that they don't get saturated too easily. So I'd say, like, in a version 2 of this, we would look for enzymes that are very easy to saturate in general. We thought about actually tried some things related to this when I was still in the lab, to try to change the system up so that we could more easily saturate the phosphatase without saturating the transcription factor here, like, for example, mutating the transcription factor to make it less strong to binding. But the difficult thing there is that we would end up pushing the system to the regime where you wouldn't be able to activate the output anymore, basically, because then the activation was too weak. And then we also have to contend with the fact that these enzymes are probably not perfect enzymes, and they're binding to this transcription factor and sequestering some of it away. So you end up kind of having a lot to deal with there. And that's another reason why we can't necessarily just reduce the Km of the enzyme, because based on the catalytic and binding rates, you would have to do it by reducing the K, or by, reducing the K off, basically, or and so you would probably get a lot of sticking of the enzyme to the transcription factor, which might also be a problem. So we think that, like, this system works pretty okay, at least as like a reasonably robust system, and something that is a good scaffold for building signaling networks, we really like. But if you wanted to get something that had this more idealized integral-like feedback control, I think it would make sense to find an enzymatic process that is very easy to saturate, but then also doesn't, through binding to the substrate, prevent it from doing any, like, functional activity that it needs in the case where it's still going to be stuck to it. So I'd definitely be interested in other architectures.
Are you aware of endogenous systems that do this, so that operate in that regime that you mentioned?
I'm not sure. I know of, like, a few feedback systems in the cell that do have this similar kind of architecture where they produce a phosphatase in response, and then that phosphatase dephosphorylates the transcription factor or some kinase upstream of it that is pushing its expression. But I'm not sure if those proteins are working in, like, an integral, like, fashion. I think maybe the closest thing would be signaling pathways like the MAPK pathway, where everything in it is a kinase or a phosphatase. And so you have direct feedback from a downstream protein back up the pathway. And I don't know if the parameters of that work out to where it's operating as an integral-like controller, but nevertheless, they get nice linear relationships between signal coming in and phosphorylation on the back end of it. So I think it's at least close, and also has the benefit of being very fast.
Mm-hmm.
Yeah. Thank you very much. And again, really nice work and nice presentation, Ross.
Yeah. And also Mustafa shouldn't be too humble here. So his lab has developed some really nice integral feedback control systems that do operate in the integral-like regime, and are thus quite robust. And you guys, did your work with, I saw that when it was on bioRxiv, but has your recent mammalian feedback controller come out in the published literature yet? Okay.
Yeah, we have one that came out in PNAS, I think a few weeks ago, where we have antisense mRNA, and then more recent one where we use inteins as a way to get the integral action. Actually, it is not so easy to get circuits that are supposed to be behaving like integrators to actually behave like integrators. And you've outlined some of the issues for phosphorylation, and some of those same issues actually we have to face. For example, saturation of the gene is one big issue. Degradation and dilution. Dilution is not such a big issue for mammalian cells, but for E. coli, one has to be careful about that. And of course, context dependence and burden come in as well. So it's doable, but it's not so, straightforward to be able to engineer those things. And so the parts have to be just right. I think the advantage of a circuit like yours is, of course, this protein-protein interaction that is very attractive because you can respond very quickly.
Right. Yeah.
But zero order reactions again are hard to engineer, although we know that nature uses them. For example, bacterial chemotaxis employs one such mechanism.
Yeah, so I hope that at some point we can get this to work out where we can tune it right so that we can get it to be more robust. But I think that it'll be it's just exciting to think about all of the new tools, all the new feedback control systems that are now coming out a bit more regularly, and the feed-forward control systems that we also worked on. All of them are going to have certain advantages and disadvantages in terms of circuit design. And so we can think about how we want to piece them together depending on the process that we want to control. Do we need to just buffer it a little bit, or does it need to be precise, and how fast does that feedback have to be? Do we need to hook this up to other systems? And so depending on all of those properties, we can think about, okay, the antisense system, this would be great for like X, and maybe phosphorylation, we can use it for something else.
Yeah, exactly. And I think in many applications, you may not need perfect adaptation. I mean, this is kind of it's interesting from an intellectual level to be able to say, okay, is it doable? And what conditions do we need it? And we know that natural systems do use perfect adaptation because we see it at various levels. But it's a fairly stringent type of regulation. And maybe in, signaling pathways, it's not necessary. So in other systems it might. But I think going forward in the field, as you pointed out, I think it would be extremely useful to have an array of many different types of controllers, some based on RNAs, some based on proteins, some based on protein-protein interactions, and so on, as each one of those will be effective in different application modes. So we do need to develop this toolbox. And I think that's something that I think as a community we ought to focus on. I mean, also for stem cell differentiation, but as well for cell therapy. It's another big area of application. You mentioned CAR T cells, where one would want these controllers to be monitoring the cytotoxicity and maybe producing antibodies that kind of try to mediate cytokine storms and things of that sort.
Right, and this is exactly something that I should have mentioned, is that one of the great benefits of differentiating T cells from stem cells is that we're no longer kind of beholden to the capacity limits that we are when we try to engineer these cells from donors. A lot of the pharmaceutical companies doing CAR T therapy are just lenting in one thing, maybe two things. There's some work now to do CRISPR knock-in to these cells, but it's not super efficient. And you're still limited in what you're probably able to put in that context. But with a stem cell, we can engineer, we can program them with many different programs. We could have developmental programs, we can have therapeutic programs. And the challenge becomes, A, can we prevent those from affecting differentiation sufficiently well so that the cells still differentiate correctly? And then B, once the cells are differentiated, can we make sure that they're still active? And so I think that those two challenges are going to be very interesting things to work on over the years, but hopefully then we can set up systems where we can put in these sophisticated gene regulatory networks, have them function in the therapeutic context, and be able to define all of it in clonal stem cell lines that you could mass manufacture. And so that's sort of like the very long-term vision of where this can go. And hopefully move, cell therapies away from just tweak the cell to, completely control the cell and create a product that, both takes advantage of the complex nature of the cell, but also buffers against the complex nature of the cell so that it doesn't do anything that we don't want it to when it goes into a tumor.
Wonderful. I love this. Actually, your conversation answered many of my questions. So, because I always, use airplane controller as a comparison system or device when I think about biological systems. But of course, to go there, there are many challenges you've already discussed. Of course, one challenge in biological systems is speed of response compared to the electronic systems speed. But you're using phosphorylation, probably smart because that's fast compared to other system. But still we are not there yet because electrical system is too fast. And another challenge you already kind of imply would be stability of each component, dilution, degradation, including protein and RNA in the circuit. Also giving lots of noise, as you know, so, and I believe you already can cover all of that, but, Ross, my question is, could you kind of comment on this point, probably a couple of sentences to summarize your perspective, potentially as a professor later, and then probably that might be your theme in the future, I guess. Could you give us, a few sentences, what is the challenges, what is your solution toward that or opportunity in the future?
Yeah, I like to think about these other kinds of systems as well, because we're very inspired by all of the mechatronic systems and electrical systems that have been designed before, and to think about how can we make biology work robustly, predictably. And one approach that people have done so far is to try to measure everything that's in the cell and then to make some predictions based on that. But what we're trying to set out to do here is to say, we want to build systems that are resilient regardless of what's going on. And we want those systems to be able to program things like cell development, to be able to program things like cell function in a therapeutic context, in a resilient and robust way, without having to know everything about what is going on, so that we can make therapies that are more effective, but also more safe. And I think that that's very important for like the next generation of cell therapies. And also cheaper, because hopefully we can make them from stem cells and just mass produce them, and we don't have to get them from individual donors. But there's a lot of challenges along the way. Specifically for cell therapies, we have to make these cells in a way that's compatible with people, and there's a lot of great work being done right now to figure out how to sort of work in this immune compatibility at the stem cell stage. And so kind of incorporating that with the sort of gene control systems that we have, we can hopefully build these systems that can be mass produced, can autonomously drive differentiation, and can build cells that are within the body autonomous little agents that can detect the specifics of being in like a tumor and then put out the correct response in accordance with what they see, all in sort of an allogeneic, which means not coming from your self donor, but from these cells that we produce in vitro. And I think that getting towards that would be like a great significant advance, and we can contribute a lot of tools towards this in the future Jones lab, so to speak, in terms of programming development and programming cell therapies with these types of control systems, and also just generally studying questions like, what are the inputs that a cell sees, and how does the cell process those inputs, and then how, when we design genetic circuits that go into the cell, do they interface with what's going on in the cell and what inputs the cell is seeing, because knowing what's going on there is going to help us to build resilient systems and to be able to program cell behavior.
Wonderful. So, I would just say, again, if you are employer, hire him. I mean, his vision is fantastic. And that's my, comment. So I guess we spent, more than, 1 hour right now. Actually, I have more questions, but I want to ask that, separate session. So I will close if I don't see any more questions. Let me double check one more time. So I didn't see question from audience, and I will ask my question later. That's a more technical question. There's no chat. Okay, I'll close in this case. Okay, so thank all for joining and staying today, especially during the vacation weeks. And we'll meet again next week on August 25, Thursday, the same time, the same Zoom link. And I will say the most exciting thing is that this seminar series will have 1 year anniversary on August 26th. Exactly, August 26th, 1 year ago, we have first seminar with the Francis Arnold and, India EBR's director, and also Doug, Biomate director, and then NSF, director, Teresa Good. And we have the wonderful time. And then since that, we have the weekly meeting. And then I'm super excited to have, 1 year anniversary. And then we will have, actually 1 day before, that is the August 25, we'll have Dr. Nigel Mossi, DOE JGI director, and Kaushik Venkatesan from University of Toronto, another Canada university, and I believe the rival university of the UBC, Vancouver. And as usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers, and I will promote you to panelists who can speak and show your handsome and pretty faces. Thank you. I will stop recording. Let me stop recording.